Let's go. That was very bad.
What did I like today? You officially got all of the
You're
first
gonna
half.
start off.
I just really want new stuff. Like, just give me
This
new
is gonna
toys.
be very
Wait,
nice.
that is not new at all.
Let's
I
just
have
adjust
the
this.
Prince and oh, here's my bookmark. Okay. Come on. Oh, it's new. It's new. Charlotte Bronte. Charlotte Bronte. I don't know. I don't know how to say that. Very excited to have something new. And this one is a rare. This is a duplicate. And based off the feeling of this one, I think we have another teacup. Yes. Okay, this feels crazy and super sad to say, but this is our capsule. I definitely should have purchased more. All I ask is that everything in here is new, and ideally we get the cat. I mean, that's a lot to ask, but yeah. Please. Oh, 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 that could have been bad. Come on. Give me something good. Okay, this is technically new, because this must be the Comet Dracula book, considering that it's so much smaller than the huge luxury one and it doesn't have a bottle of blood or whatever in it. And, oh, it's also new! Wait, this is going really well. I really wanted this one. This is the Little Women book. I'm so happy about this. This is great. If this last book could also be new, that would just be like the cherry on top. That would be the oh my gosh, wait. It's new. I know it looks a lot like this one that we've gotten like a billion times, but it's not. It's actually different. Like everything was new from this last capsule, but I guess we still have to check what this other item is here. This one is also a rare. Oh no. Based off the feeling, it must be a teacup. It must be a teacup. It is.
Used to monitor
Oh, this is
bacterial
so much
transformation with
paperwork.
foreign
Oh,
DNA.
this series two books
Nuclear
covered in
membrane is found
shells.
in
I want to go back to
eukaryotes.
the
No
mini pages
organelles
of
like the
series
ones in
one.
eukaryotes are
Those
found
shelves
in
are so
prokaryotic
incredible.
cells,
So
except for
thanks.
some
So let's stuff
ribosomes.
these shelves with goodness from series one. Because all these
And
experiments are
nuclear
so
membrane found in
cool, but
eukaryotes.
hopefully
No
not in
organelles like the ones in
yours in series
eukaryotes
two, at
are
least that I
found in
found. Yours
prokaryotic
was a little
cells,
sad,
except for
but
ribosomes.
oh. And wow,
Prokaryotes
these
have
shelves are fully
something
stocked
unique in the
with
form of
just the mini
inclusions.
brand
A
books and
specialized
fragments. So it
differentiated
just looks
form of
so
cell membrane
incredible
called
and beautiful,
mesosome
but
is a
we'll have to put it
characteristic
together. Now, the one
of
thought that I did have,
prokaryotes.
we did get
There are
cards in
essentially
here,
enfoldings of
but
cell
they don't
membrane,
have anything in them.
cell envelope,
So
and its
spice.
modification.
So to spice
Most
up the vibe a little
prokaryotic
bit,
cells,
I'm gonna use
particularly
some
the
leftover
bacterial
bread that
cells,
I
have a
have
chemically
from mini
complex cell
words and
envelope.
carefully pour
The
some
cell envelope
into the
consists
cup.
of a
But
tightly
like
bound
Shira, to
three-layered
me it looks there's
structure.
actually some
The
tea dropping in
outermost
there. Like I saw the
glycocalyx,
red version
followed by
of
the
the
cell
cup to
wall,
make it different
and
considering the tea
then the
like lemon
plasma
tea or
membrane.
something. In this one,
Although
it's in
each
a
layer of the
brown and
envelope
white envelope,
performs
and it's
distinct functions,
really warm. What is the
they
smell?
act together
Oh,
as a
these
single
look so much cute.
protective
I think these are
unit.
100 times more fun now that we have
Bacteria
like
can be
simple
classified into two
drinks inside of
groups
them.
on the
But
basis of the
candles, because we
differences
have
in
a
the
billion of
cell
these to
envelopes
experiment with.
and the
They
manner
don't
in
have
which
like
they
a huge
respond to
thermoplate
the
top view
staining
and kind of
procedure
dripping down the
developed by
sides to
gram
like
vis.
melting wax. I also use
Those
a
that
salt
make
liquid
up the gram
manually.
stain are
I
gram
use darker
positive,
to color them
and
black,
the
and then I use a
others
small
that do
amount
not
of
are
orange
called
chalk
gram
glue to
negative
make up the
bacteria.
flame to make the candles a little
Glycocalyx
more
differs in
realistic. And there's also
composition
like
and
different
thickness
color. And
among
yeah, the mini
different
words book
bacteria. It
series.
could be a loose sheet called a slime layer in some, while at another it may be thick and tough, called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is a selectively permeable membrane in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extensions of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration, secretion processes to increase the surface area of plasma membrane and enzymatic content. And in some prokaryotes like cyanobacteria, There are other membranous extensions into the cytoplasm called chromatophores, which are pigments or which contain pigments. Bacterial cells may be motile or non motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria, but do not play a role in motility. The pili are elongated tubular structures made of special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks and streams and also to the host tissues. Alright. Now, ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometers by 20 nanometers in size and are made up of two subunits, 50s and 30s units, which when present together form 70s unit. Prokaryotic ribosomes are of 70s. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA, messenger RNA, and form a chain called polyribosome or polysome. The ribosome of a polysome translates the mRNA into proteins. Inclusion bodies. Reserved material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granules, cyanophycin granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. Now, let's understand this one by one. Stream. Stream. Now let's understand this one by one. पढ़ रहा हूँ ना ये सब ओके पढ़ रहा हूँ अच्छा मुहार तो मेरा बच्चा प्लीज तुम इनको सपोर्ट करो हिबा सपोर्ट करी हिबा look at me when I'm talking to you support करी उनको. Yes, now let's understand this one by one. In prokaryotes, the cell has a naked genome, or it lacks the nuclear membrane. And since it lacks the nuclear membrane, what we can say is that the lacking of the nuclear membrane will have shown that the DNA is, open. There are also small DNA circle structures known as plasmids. These are having some phenotypic characteristics which are capable of showing a resistance to the antibiotics. And by showing some resistance to the antibiotics, we study this in the topic of class 12 biotech. Now, they are also capable of known as genome taxi because they are used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. Alright, these nuclear membranes are the ones that are found in the eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cells. So what I mean here is that prokaryotic cells are having many different kinds of structures. One is known as the mesosome, and the other one is the inclusion bodies. Mesosomes are the enfoldings found in the cell membrane. This could be of a tubule, a vesicle, or a lamella. The inclusion bodies that we are going to study are some kind of reserve material in the prokaryotic cells that will be stored in the cytoplasm. Now, let's try to understand the cell envelope. The cell envelope has overall three layers. The first layer, or the most outside layer, is the glycocalyx, or a sugar coating layer. This glycocalyx layer are of two types. It could be a kind of layer which is very smooth, and this kind of layer is known as slimy layer. The other one is known as the tough, thick layer that is known as the capsule. Inside the glycocalyx, we have the cell wall. This cell wall is determining the rigidity and turgidity of a cell. They're capable of giving a shape to that of the cell and providing even the structural support. It is also capable of having some resistance against the process of plasmolysis. That is, if it goes against a lot of osmosis here, then it can prevent the bursting or collapsing of a cell. The plasma membrane is the one important kind of a structure in the cell, which is capable of having the transfer of materials through the process of diffusion and osmosis. The diffusion can be of many types, that is facilitated diffusion, carried protein diffusion, and we're going to look at that. Now, we are going to understand mainly about mesosomes. Mesosomes are of three types: a vesicle, tubule, and a lamellae. They help in the cell wall formation, DNA replication, and distribution to daughter cells. How do they help in cell wall? Well, the material will be carried to this vesicle, and this vesicle will store that material into the cell wall, in the formation of cell wall. DNA replication? Well, the DNA replication will be done by tubules. What it does is that it will take that DNA and separate it into two. It also helps in the distribution to two daughter cells, meaning it can help in cytokinesis. That is in the other chapter. This thing will help in separating the cell, two daughter cells. And if you look at the processes like lamellae, this is lamellae. What it will do is it will help in respiration, secretion processes, and even increase the surface area of the plasma membrane and its enzymatic content. There are some enzymes that are present and they act as a biocatalyst. We all know that, right? I'm not sure, but yeah. In some bacteria or in some prokaryotes, we see there are like kinds of cyanobacteria. There are the other membranous extensions into the cytoplasm called chromatophores. Now, what is this chromatophore? The chromatophore you see is again an unfolding of the membrane. It has pigments present, a colored pigments, like a color that you can see in a rainbow. That kind of color pigments can be seen in chromatophore of the prokaryotic cell, or mainly specifically bacterial cell. The bacterial cell may be motile or non-motile. That means it can either move or they may not move. And if they have a thin filamentous extension from the cell wall called flagella, then it will help in their motility of the cell. So they will show some range in number and arrangement of flagella. It is of three types: filament, hook, and basal body. The filament is the longest part or the longest portion and extends from the cell surface to the outside and from the what we can say is cell wall, from the cell wall. The other two are pili and fimbriae. The pili are elongated tubular structures made of a special protein connecting the cells, and fimbriae are small bristle-like structures which are found on the walls of the cell. They are known to help attach to the bacteria to rocks and streams, and also to the host tissue. So this helps in the attachment of on a particular surface for that bacteria. Now let's understand this ribosomes and inclusion bodies. Ribosomes are of many types. They are the universal cell organelle that can be found in many different types of cells. They are associated with the plasma membrane, and they're about 15 to 20 nanometers in size. They are made up of two subunits, 50S and 30S, which are present together form 70S prokaryotic ribosomes. Ribosomes are a site of protein synthesis, and several ribosomes may attach to form a single mRNA and form a chain called polysome or polyribosome. The ribosomes of a polysome translate the mRNA into proteins. Alright, that's the main thing we should know. That there is something called messenger RNA or mRNA that can actually form a chain. It can form multiple mRNA chain here, and this is known as polyribosome or polysome. The ribosomes of a polysome translate this, and this translates this mRNA into the proteins. Now let's look at something called inclusion bodies. These are some kinds of reserve materials that are found in the cytoplasm of the prokaryotic cells. These could be of, phosphate granules, cyanophycine granules, and glycogen granules, or even gas vacuoles. They cannot be bounded by any membrane system and live free in the cytoplasm. It shows that it is a non-membrane bounded organ. These are just examples to go through. It's better to learn them while going through or by reading them. Now, the next one is eukaryotics. The eukaryotes includes all the protists, plants, animals, and fungi. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition of eukaryotic cells, they have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possesses cell walls and plastids, and a large central vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are absent in the plant cells. By looking at this, we get to know that there's another kind of cell known as eukaryotic cells which have a definite nuclear membrane bounded nucleus, and this nucleus we see is typically present in eukaryote cells. Now, eukaryotes can be seen in all the four kingdoms, that is, protista, plants, animals, and fungi. Also, by looking at this, we get to know that it has an organized nucleus with a nuclear envelope, and it also has a variety of complex locomotory and cytoskeletal structures, showing that how big the cell of eukaryotes can be, and it well, could be of very different types here. Plant and animal cells are different as form possess cell walls, plastids, and large central vacuoles which are absent in animal cells. There are of very small vacuoles that can be found in an animal cell. By looking at this one here, we get to know that the centrioles are present in animal and are absent in this. This is the diagram given for the plant cell, and this is the diagram for given for the animal cell. Like yeah, 10 बजे साढ़े 10 and this is the diagram for animal cell. This is the diagram for plant cell. This is a cell wall, and this is the cell membrane, if you can see through this. And here, this is your full cell of animal, and you can see that this is a 9 standard textbook diagram. This is all the overall analysis that you can get from what you know here. Now let's talk about this cell membrane. Cell membrane is very unique. It is made of lipid phosphates. After the advent of electron microscope, that is the discovery of electron microscopes in the 1950s, or used or the application of this, the chemical studies on cell membrane were made, especially for RBCs, enabling these scientists to deduce the possible structures of the plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. Okay, you can look at the cell membrane and you get to know that lipids and proteins are here. These proteins are our carrier proteins, or peripheral proteins, or integral proteins. This is your lipids that can be seen here, and this two layers of lipid is known as lipid bilayer. The major lipids that can be found here is phospholipids, and that are arranged in a bilayer. Also, the lipids are arranged within the membrane with a polar head. They have a polar head, and if we look at this, Their tail is hydrophobic, which means that they are against the water. They do not want the water to be in contact. That's why the tails are inside. And the head is hydrophilic, means they love water. And due to the loving part of water, they interact with the water from the outside environment or inside environment. Now let's look at this and get some small ideas of fear. That hydrophobic tilts towards the inner part this ensures that the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. The phospholipids membrane also contains cholesterol. These small structures you see are known as cholesterols, which are the membrane stabilizers. Biochemical investigations clearly reveal that cell membranes also possess protein and carbohydrates. There are carbohydrates, and I think these represent the carbohydrates here, and the proteins are these structures that you see. There are two types, a peripheral protein and an integral protein. This peripheral protein is this part, and the integral protein is this inside part here. Let's look at this and get the idea of what is exactly what we are talking about. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocyte has approximately 52% of proteins and 40% lipids. Depending on the ease of extraction, membrane proteins can be classified as integral, peripheral proteins lie on the surface. These are the peripheral proteins that lie on the surface of the cell membrane and of this membrane, while the integral proteins they are partially or totally buried in this membrane. The fluid mosaic model of plasma membrane shows that these proteins are partially or totally buried in this cell membrane or this lipid bilayer. That is what exactly it's mentioning. Now, let's look at this part here. An improved model of the structure of cell membrane was proposed by Singer and Nicholson, 1972, widely accepted as fluid mosaic model. The quasi-fluid nature of lipid enables the lateral movement of proteins within the overall bilayer, showing that the quasi-fluid nature, showing the some ionic nature, even the covalent nature of this lipid bilayer, enables the lateral movement of proteins. It shows that how it can show the movement of proteins, and we're gonna learn this through this part here, given in this paragraph. So there's another thing is that the ability to move within the membrane is measured as its fluidity, because there's fluidity is the ability to flow. Fluid nature of the membrane is important for the point of view of functions like cell growth and formation of intercellular junctions, secretions, endocytosis, cell division, etc., etc. One of the most important functions of the plasma membrane is the transport to the molecules across it. The membrane is a selectively permeable membrane. Since it is a selectively permeable membrane, to some molecules that are present on either side of it, that could be outside or inside of the membrane, these molecules can be transported to many types. This could be a normal transport known as passive transport following the normal concentration gradient, which we're going to talk about it. Many molecules can highly briefly come across the membrane without any requirement of energy, and this is called as passive transport. Suppose if there is high concentration gradient here and there is low concentration gradient. So this higher concentration gradient tends to move to this side without any involvement of energy. And due to that no involvement of energy, we're calling this as passive transport. Passive transport is obeying the concentration gradient. This is done by the process of a simple diffusion and moving along the concentration from higher to lower. Water may also move across this membrane from higher to lower concentration, and this is known as osmosis. As a polar molecules, cannot pass through the nonpolar lipid bilayer, they require a carrier protein. Polar molecules have charge in them. And since they are not capable of passing through this non-lipid bilayer as they are charged, these require these carrier proteins. These are known as carrier proteins. They take this charged molecules from here to here or here to here. This is how the facilitated diffusion is taking place through this one, where we get to know that the carrier protein is basically of the membrane or to facilitate their transport all across the membrane. And this, if few ions or molecules are transported across this membrane against their concentration gradient, now from that is to lower to higher concentration, which is against the concentration gradient, they require energy for that. In order to acquire energy, they need to utilize the ATP, that is adenosine triphosphate. And that's how the active transport comes in. The active transport of these molecules is done from lower to higher concentration, disobeying the concentration gradient. This is because some amount of energy is involved, it is an energy dependent process, and the ATP is being utilized. Therefore, this is an active process or an active transport. This is sodium plus and K plus pump can be seen through this. This is how your overall ionic molecules get transported. Now let's talk about cell wall. As you may recall, a non-living rigid structure is known as cell wall. These are rigid, they provide shape to the cell, structural support to the cell. They are outer covering of the plasma membrane of fungi and plants, but not for animals though. Cell wall not only gives the shape to the cell, they protect the cell from mechanical damages and infections. It might also help the cell to have a cell-to-cell interaction and provide barrier to undesirable macromolecules. Algae have cell wall made of cellulose, gelatin, amines, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and proteins. It describes what all kinds of different things you can see through these macromolecules passing through that specific organism, like say algae, which is a plant, have cell wall. Okay, having cell wall is because it's a plant or a lower plant. It is made of this cellulose, skeletons, minerals, and minerals, showing that this is how the cell wall is made of. And even it is other plants, it is consisting mainly of cellulose, some amount of hemicellulose, pectins, and even proteins. We looked at how the proteins are involved too. The cell wall of a young plant cell is a primary wall and is capable of the growth. They are since capable of the growth. These are gradually diminishing as the cell matures and the secondary wall is formed on the inner towards the membrane side of the cell. The middle, showing that this is the primary wall capable of the growth, gradually diminishes as the cell matures, meaning that this kind of, during the maturation of the cell, when the cell differentiates, it stops dividing. Then this will become steeper and thinner, and that's how your maturation fully takes place in that cell. Secondary walls. Okay. The middle lamella is a layer mainly consisting of the calcium pectate, which holds the glues the different neighboring cells together. The cell wall and middle lamellae may be transferred by plasmodesmata. This, you see, guys, is plasmodesmata. This is your two cells being connected by plasmodesmata. And on the layers of plasmodesmata, you find lamellae. The middle lamellae may be transversed by the plasmodesmata, which is connected to the cytoplasm of neighboring cells. That is how we get to know the overall topic. Now let's talk about endomembrane system. What exactly endomembrane system is? Which each with each of the membranous organelles is distinct in terms of its structure and function. Many of these are considered together as endomembrane system because their functions are coordinated. They function together, and due to that togetherness function, these endomembrane system consists of endoplasmic reticulum, that is Golgi complex, lysosomes, and vacuoles. The function of the mitochondria, chloroplast, and peroxisomes are not coordinated because they have very different functions to that of what normally these cell organelles do in a connected way. Now let's understand the first cell organelle, that is endoplasmic reticulum. These are some kinds of organelles that are attached to the outer nuclear membrane. This, you see, guys, is your endoplasmic reticulum. You also see ribosomes covering some parts of the endoplasmic reticulum, and this is known as rough endoplasmic reticulum, while this part is known as smooth endoplasmic reticulum. Electron microscopic studies of eukaryotic cell reveals the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm. This is known as ER, and ER divides the intercellular space into two distinct compartments: luminal inside the ER and extraluminal cytoplasm compartments, showing that internal luminal and external luminal, and this external part of the part here is fully cytoplasm of the cell. ER often shows ribosome attached to the outer surface. This is the ribosome attached to this outer surface of the endoplasmic reticulum, and this is called as rough endoplasmic reticulum, RER. And if there is an absence of ribosomes, where there is no ribosomes here, then these are known as smooth endoplasmic reticulum, SER. So this is RER, and this is your SER. That's what you can say. See, RER, SER. Now let's look at some ideas here. That RER is frequently observed in cells actively involved in protein synthesis and secretion. They're extensive and continuous with the outer membrane of the nucleus, because since the ribosomes are involved in protein synthesis, that's functional. We get to know about this idea, and even helps in secretion of uncertain substances that we'll see later on. And the smooth endoplasmic reticulum of the is the major, steroidal. It can is the major lipid-like producing steroidal hormones, which are synthesized in SCR, or smooth endoplasmic reticulum here. That is what exactly here is saying. That can synthesize the lipids, and it is in animal cells. It's like lipids, like steroidal hormones, which are synthesized in SCR. Now we have Golgi apparatus. The Golgi apparatus was discovered by Camillo Golgi, first observed two densely stained reticular structures and nearly the nucleus. This looks like normally we draw Golgi structures as a Wi-Fi structure, and this, you see, guys, is a bit confusing. We always confuse with what is convex and what is concave shape, and we should get to know this as soon as possible. Then we'll move on to understand what exactly this topic is talking about, how is the function taking place, then the other two uh, cell organelles. Okay, now Golgi apparatus. Gamilogolgi observed this and sustained. Yes, they consist of many flat disc-shaped like sac structures, or known as cisternae, and these are of at least 0.5 to 1 micrometer. These are stacked parallel to each other. They are not different. They are stacking. They are in stacks of parallel. And looking at this, we get to know that there are varied number of cisternae present in the GC, that is Golgi complex, and cisternae are concentrically arranged near the nucleus. The more of these Golgi complex in cisternaes, or these you see are cisternaes, they are concentrically, they are more in number near to the nucleus. The Golgi near to the nucleus with distinct convex cis, or the forming phase, and the concave trans, or the maturing phase. By looking at this forming phase, this is your convex phase or cis phase, the lengthiest part, also known as forming phase, while the other one is the maturing phase, where this part you see, the smallest part you see, is your concave. and this is known as your trans or maturing cell.
I have two of the newest series
Now
from
let's move on
Syllabus. This
to
is the
understanding that a principle for functions performed by the Golgi complex. It helps in the packaging, modification, and even in the transportation of a substance. So it packages certain materials, deliver it to the intercellular targets, or that is inside the cell. These or cell organelles are given that product which is packaged by the Golgi apparatus, and materials to be packaged in the form of vesicles from the ER fused with the cis phase of the Golgi apparatus. So, due to the structure of the vesicles, which are fused from the endoplasmic reticulum, with the cis phase, what is cis phase? Convex phase. This is your convex cis phase of the Golgi apparatus that is fused with the endoplasmic reticulum towards about the vesicle, okay, of the ER, and it moves towards the maturing phase. Move towards the maturing phase is this is your maturing phase. It moves towards it. This explains why the Golgi apparatus is close in association with the endoplasmic reticulum (ER). Number of proteins is synthesized by ribosomes on the endoplasmic reticulum. Okay, you can say that the many proteins synthesized by ribosomes because ribosomes is a protein synthesizing substance or a very small particle. And on the endoplasmic reticulum, these are modified in the cisternae or of the Golgi apparatus before they are released from its trans phase. What is trans phase? Concave trans. This is your concave phase. It is important site of the formation of glycoproteins and glycolipids. This is the important one. Glycoproteins and glycolipids and even the lysosomes. Yeah. These are also capable of forming something called, lysosomes. Okay, they are made by lysosomes are suicidal bags of the cell, and these suicidal bags of the cells are originally made by this Golgi complex. So what exactly is happening is that the protein and lipid that is coming from the ER will go through these cisternae one by one, and from this protein gets converted into glycoprotein, and this lipid is also going through this same phase where it moves from the cis phase to the trans phase, and this phase, moving to trans phase, we get to know that this lipid is converted into glycolipid. That is what exactly it's mentioning here. Now, as I said, the Golgi complex is capable of making lysosomes. Now, what are lysosomes? Lysosomes are basically your membrane bound vascular structures. They are known as your suicidal bags of the cell. And by the packaging the Golgi apparatus, the isolated lysosomal somal vesicles, they have been found in very, very rich type of hydrolytic enzymes called hydrolases, lipases, proteases, carbohydrates, carbohydrates, and nuclease. That is your hydrolases enzyme. Hydrolases enzyme can be seen in a chapter known as biomolecules. This is your hydrolases enzyme. The hydrolases enzyme is basically catalyzing a reaction of breaking the bonds, like this carbon-carbon bond, C halide bond, which is not there for now, but PN bonds, phosphorus and nitrogen bond, then ester peptide bonds, and there's another one, glycosidic bonds, that is mainly found in our biomolecules chapter. Now, moving on back to where we were, is the main idea of the lysosomes, that these have very highly enzymatic content inside them. These are like enzymes of hydrolases that catalyze a reaction of hydrolysis. That is lipases, proteases, carbohydrates, and even nuclease. They are active at the acidic pH because enzymes are affected by this factor of pH and temperature. They are capable of digesting carbohydrates, proteins, nucleic acids, and lipids, as due to these enzymes that are found here. Now, talking about the vacuoles, that is the tonoplasts. That is, they contain a membrane-bound space known as tonoplast. This is your tonoplast. This membrane you see is a tonoplast membrane that can be found in the cytoplasm. They contain water sap, excretory products, and other useful material, not useful materials, my bad, not useful materials for the cell, which can be used later on if there is any kind of important things that might go on. But mainly, it's not useful. It's not necessary for the cell. The vacuole is bound by a single membrane called tonoplast, as I've mentioned. Plant cells, vacuoles overall occupy up to very large amount of the cell or the volume of the cell, and due to that, we look at this and get the idea. Oh yeah, that's for real because the volume of the cell is mainly covered by this vacuole, and 90% of it, that is that shows or signifies that plant has very large vacuoles, whereas animal vacuoles are very small. In amoeba we got some contractile vacuoles, which is important for osmoregulation and excretion. This is this it stores those kinds of stuff, which is having those kinds of similar functions here. And in protists, food vacuoles are formed by engulfing the food particles. So there are many vacuoles that have some very special functions, unique functions. You may have studied about food vacuoles in class 10th, how that it digests the food and then it adjusts the waste out, the undigested waste. Now, the main very important topic, mitochondria. Everyone knows that mitochondria is the powerhouse of the cell. Nobody even forgets about this cell organelle. This cell organelle is even more famous than that of the normal cell, I believe. Mitochondria is known as the mitochondrion in singular form, and they can be specifically stained by Janus green stain. These are easily not visible under the microscope, uh, because you cannot normally see them, but once you stain them up with this Janus green stain, you'll find out that yes, these can be seen in the microscope. Number of mitochondria per cell is variable because it can be different based on what we actually observe, and the physiological activity of the cells can be functioned here and can be seen that in terms of the shape and size. Also, there is a considerable degree of variability that can be observed, and typically this is your mitochondria, which is sausage shell shaped cell or cylindrical in shape, having the diameter as 0.2 to 1 micrometer average. And we can say that the length is overall like 1 to 4.1 micrometer. Each mitochondria is a double membrane bounded organelle, and this double membrane bound organelle, that the outer membrane bound is, can be seen here, which is it feels like it is blown up, like it's very much having a lot of thickness inside of it. That's how the outer membrane looks like, but the inner membrane is innerly folded due to, because it increases the area for ATP production, which we know that through the process of ETS, which is given in the other chapters. But here we just need to know that it helps in the production of ATP by increasing the surface area of the mitochondria. Now, understanding this, that the inner compartment is filled with dense homogeneous substance known as matrix of the mitochondria. The outer membrane also forms a continuous limiting body and boundary of the organelle, but this main thing, inner membrane, forms a number of enfoldings, and these are called cristae. In Golgi complex, we call this as cisternae, not cristae. We call this as cisternae, and here we call this as cristae. We mainly get confused with the names, so we need to be really careful based on that. Now, understanding this, the two membranes can have their own specific enzymes here, okay? These which we go through this ones by many kinds, like there is hexokinase, no, not hexokinase, there is isomerase, and there is another enzyme like pyruvic dehydrogenase. Many enzymes can be seen, and those aren't mentioned here, but yeah, it's there in the future chapters. If you study deeply, you'll get to know. They produce cellular energy in the form of ADP, and they are called as the powerhouse of the cell. Very popular known line that almost everyone knows. The matrix also possesses single circular DNA molecule. This single circular DNA molecule can be seen along with some amount of RNA molecules. These ribosomes that you see are present in this cell organelle, but the only thing is that instead these ribosomes are not of 80s, they are of 70s ribosomes. The components required for the synthesis of proteins, mitochondria, are divided by the process of fission, which is an asexual reproduction method. But here, what we need to know is that fission is not only about asexual reproduction, it's breaking down of that one substance to in multiple substances that are very small. That's how fission mainly means. There's another word known as fusion, where different substances come together to form a big one substance. That is different. That is opposite to that of fission. Now, now let's talk about something related to mitochondria, that is plastids. These are also very unique, but they can only be found in plants. These are found in plants and euglenoids, and euglena are, I think, generally found in the kingdom Protista. Kingdom Protista, you find these euglenoids. They are mainly observed under the microscope as they are very large. They bear some amount of pigment, imparting specific colors to the plants. And these are of three types based on giving the color, that is chloroplast, chromoplast, and leucoplast. The chloroplast here is capable of giving a green color to the plant, providing chlorophyll pigment to the plant too. And chromoplast is another pigment that gives another pigments like carotenoid pigment, carotene pigment, xanthophyll pigments too. And this fat soluble carotenoid pigment gives the orange yellowish color, by the way. Leucoplasts are colorless or white colored plastids that have the function of making storing of a certain nutrients, like it can be known as amyloplast because it stores carbon dioxide. It is known as elioplast because it stores fats, and it is not only that, it is known as aleuroplast because it is capable of storing proteins. Now, this after that, we study mainly about chloroplast because chloroplast is very much important in plant physiology chapter. So we gotta go through this once for and for all, complete the whole thing. So the majority of the chloroplasts that you see are of the green plants that are formed in the mesophyll cells. These mesophyll cells are the leaves that are lens shaped, oval, spherical, discoid, or even ribbon like organelles that have even like organelles having variable length as 5 to 10 micrometers and width as 2 to 4 micrometers. Their number varies from one per cell of the chlamydomonas, a green alga, to 20 to 40 per cell in a mesophyll. Okay, now this shows that chloroplasts can be in many different numbers, and showing that number. Okay, we are close, guys. We are super close. We actually completed. We are making a lot of progress. Not gonna lie, I thought we were going slow, but here we can look at the progress that we have overall made. So the progress here is now we are at chloroplasts. After chloroplasts, we are gonna go through some small topics like ribosomes again, cytoskeleton, and cilia and flagella topic, centrosome centrioles topic, the nucleus chromosomes topic, and last but not the least, the small topic known as microbodies, which is only of two lines, and that's it. This chapter is very conceptual but very interesting at the same time. Now let's go through this. These are of green plants, found in green plants in mesophyll cells, especially of the leaves. They are lens-shaped, oval, discoid, and or even ribble-like organelles. These are having variable lengths as 5 to 10 micro to 2 to 4 micrometers here. There are a number of and widths as 2 to 4 micrometers here. Length as 1 to 4.1 micrometers, and this is 0.02. How much is this one? 0.2 to 1 micrometer here. Okay, so that's it given here. Hmm. All right. Like mitochondria, the chloroplasts are also double membrane bounded organelles, And of the two inner chloroplasts, wait a minute, guys. Wait a minute. Yes, my bad. I had to reconnect. There was something going on. It has been like, very great journey, guys. Like, I've been going through lots of work. We're super close, super close. There's this topic, and we're gonna read this. You know what, guys? I'm not gonna explain now. I'm gonna read this carefully, and we're gonna complete it. We're gonna wind up. That's it. So let's start. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, in the inner chloroplast membrane is relatively less permeable. The space limited by the less permeable space limited by the inner membrane of the chloroplast is called stroma. A number of organized flattened membranous sacs are called as the thylakoids, which are present in the stroma. Thylakoids are arranged in track like a pile of coins, known as grana or a singular granum. In the intergranal thylakoids, in addition, there are flat membranous tubules called the stroma lamellae connecting to the thylakoids of the different grana. The membrane of the thylakoids encloses space called lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in thylakoids. Ribosomes of the chloroplasts. Okay, yeah, the stroma of the chlorophyll contains. Okay. Oh, I did not know that. I didn't need to know that too. Where was this given? Was this given? Which could be the two enzymes here. There should be some enzyme, right? They are composed of RNA, ribonucleic acids, and proteins and are not surrounded by any membrane. Okay. Guys, I'm reading this in my mind. Do you mind? Because my throat is becoming so dry. I have to drink water. I know that's a silly reason, but then I gotta go to sleep as early as possible. I don't know why. I'm also very tired. I am having a headache. Hmm. Okay, you're close, guys. Let's go through this. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material was the nuclear stain was done by the basic dyes, was again named chromatin by Fleming. The interface nucleus of a cell when it did not divide had chromatin material. It has an elaborate nuclear protein fibers called chromatin. The nuclear matrix. This nuclear protein is known as chromatin, nuclear matrix, and one or more spherical bodies called nucleoli. Nuclear membrane was also there, will be there for eukaryotes. Electron microscopy have revealed that nuclear envelope, which consists of two parallel membrane, um, with a space between 10 to 15 nanometers between them, are called perinuclear space. Uh, Perinuclear space, yes. Here we go, parallel membranes. If I'm seeing anything, I feel like I missed something, that's why. Chicken. Yeah, inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum. It also bears new ribosomes on it at a number of the places. The nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membranes. These nuclear pores are the passages through which the movement of RNA and protein molecules can take place in both directions between the nucleus and cytoplasm. Now, normally there is only one nucleus per cell, uh, and the number of nuclei are frequently observed. Your coconut's endosperm has more than one nucleus cell, but that is given in the second next chapter. Some mature cells can even lack nucleus. Erythrocytes of many mammal ancestors. Oh yes, these are known as ghost cells. But yeah, they have some amount of properties. I guess that's what I can say. Yeah, oh my bad. The nuclear matrix of the nucleoplasm contains nucleolus and chromatin material, and also the semi-fluid inside the nucleus. The nucleoli are spherical of structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm, and it is not a membrane bound structure. It is a site for active ribosomal RNA synthesis. That is a rRNA factory. So factory of rRNA is your nucleolus. It helps in rRNA synthesis or ribosomal RNA synthesis. Okay, rRNA synthesis here. Let's go through this interface nuclear. Okay. Mm. 9 plus 0 element. Okay. Look at the chromosomes. These are done only through the different stages of cell division. In place of nucleus, chromatin contains DNA and some basic proteins called histones. Okay, and some non-histone proteins and also RNA. A single human cell DNA, some basic histones, non-histone proteins, and RNA. DNA is also there. So yeah, the single human cell DNA is overall 2 meters long. Thread of DNA distributed among its 46, 23 pairs of chromosomes. We'll study the details of DNA packaging in the form of FA chromosome in class 12th. Hmm. Okay, maybe biotech or biology. Yeah. It concerns whether it is primary construction or the centromere that is just given here as centromere. Then it's the two chromatids of a chromosome. Mm, Based on the position of the centromere, it could be metacentric, submetacentric, acrocentric, and telocentric. Metacentric of the chromosome has middle centromere. Yeah, and forming two equal arms, and of the submetacentric have it is placed a bit slightly away from that of the middle of the line, middle of the chromosome. That is at one short arm and one long arm. Then till acrocentric is that it is very much away from the centromere of the middle of the chromosome, and therefore it's extremely smaller. The upper arms are extremely smaller than that of the long arms. Then telocentric chromosomes have their terminal of their terminal centromere. Okay, you got this one idea. Sometimes if chromosomes have non-staining secondary constructions at a constant location, this gives appearance a small fragment called satellite, and many membrane-bound minute vesicles called microbodies that contain various enzymes which are present both in plants and animals. Okay. This chapter was easy, it was conceptual, but there were so many things to know from this NCRD. That's how this chapter was. So yeah, overall, this chapter is easy. It's not at all hard, but then you gotta like be really careful with what you're gonna learn. That's the thing. And yes, guys, we're overall done it. We have officially completed two chapters of biology nicely. Um, yes, we did it. Day 55 was productive. It was great. My head was aching super bad, and here you can see my head was aching super bad. And now I think I'm capable of like going normal. And yes, that was a great journey. We studied so much. D55 has ended. Only 5 days are left, guys. Only 5 days. You can do a lot of things on this 5 days. So tomorrow I might have to go to school, but trust me, during these 5 days, most of the time I will be on holidays. So I will have to be fully locked in. I'll see. And so there's another challenge known as Super 21 challenge that I'm feeling to talk about sooner. And Super 21 challenge is the challenge where we're going to do this during exams. It's a very vigorous challenge where we have to do streaming with very high productivity. Very, very high productivity. That's how you gotta do it. So yeah. And yes guys, it was great. Overall it was really great. I loved it. My face looks very bad. But yeah, trust me, bro. It was done overall nicely. And yeah, I think it's time to say bye. It was so fun. It was so fun. Bye guys. Meet you next time. Okay?